everybody. Welcome to Week 8 of Virology Part 1. I'm your Professor Vincent Racaniello, and this week we'll be tackling the topic of transcription, and that is the synthesis of mRNA from DNA templates. So this is session number 1, where we will be talking about some general principles involved in this process. Here's a wonderful electron micrograph illustrating the transcription of a gene in process. And what we have here are individual strands of DNA. They're the thicker black lines. And each of these DNAs is in the process of being transcribed. That is, mRNA or some sort of mRNA is being RNA is being made. Could be ribosomal RNA by enzymes moving down the DNA template. And you can see there are lots of short chains coming off the DNA because there are many polymerases riding down this nucleic acid making RNAs. There are 3 different kinds of eukaryotic RNA polymerases, and each of them make different types of cellular and viral RNAs, and they're called RNA polymerase 1, 2, and 3, and we'll be abbreviating them Pol 1, 2, and 3. And in this week, we'll mainly be talking about RNA polymerase 2. As you can see in this table, RNA polymerase 1 in the cell makes pre-ribosomal RNAs. They're called pre because they're subsequently processed, and those, of course, are the RNAs that go into building a ribosome. And as far as we know, there are no viral RNAs produced by RNA polymerase 1. RNA polymerase 2 in cells makes pre-messenger RNA, and it also makes pre-microRNAs and small nuclear RNAs, and does similar things for viruses, making pre-mRNAs, mRNAs, pre-microRNAs. Also replicates the genome of hepatitis delta virus. And finally, RNA polymerase 3 is responsible for the synthesis of pre-tRNAs, 5S ribosomal RNA, and a small nuclear RNA called U6. And it's also responsible for the production of the VA RNAs of adenovirus and the EBER RNAs of Epstein-Barr viruses. But as I said, we'll mainly talk about Pol 2 today. Here's an overview of the entire process that we'll be talking about this week. We're actually talking about not only transcription, but processing of the mRNAs that are produced by transcription. And we start at the very top here with a double-stranded DNA template, which is the template for transcription. Those viruses that have single-stranded DNA genomes, they have to be made double-stranded before transcription can occur. Now, we define transcription as the process of making mRNA from a DNA template. That's a very specific definition. Some use the word transcription to talk about the production of mRNAs from an RNA template, which occurs in RNA virus-infected cells, as we have talked about before. And that's fine to use that, but I just want you to know that in our course, in our textbook, if you use it, transcription has a very specific meaning. So here we have a double-stranded DNA template. Transcription initiates here, shown by the red arrow. We will always, by convention, use a red arrow to show the site of initiation of transcription. The mRNAs that are produced are capped at the 5' end. We'll talk about what that means. They are produced as pre-mRNAs. Those pre-mRNAs are then spliced. They undergo a process called mRNA splicing, by which intervening sequences called introns, and shown in pink in this diagram, are removed by a series of cleavage and ligation reactions. So the splicing is shown here. resulting in a mRNA. The 3' end of the mRNA is also polyadenylated, that is, there are a stretch of poly A residues added. And then finally, the mature RNA is exported from the nucleus. Eventually, of course, those mRNAs are used to make proteins by the process of translation, and eventually they're degraded. All mRNAs eventually will be degraded, and how long that takes depends on the specific mRNA. Some viral mRNAs are not spliced. Nevertheless, they can be exported without splicing. That is, they contain the intron. And they are also translated and degraded as usual. We'll talk about how that happens as well. Here are the steps in transcription, much like the steps in DNA replication. Promoter recognition, pre-initiation complex formation, initiation of transcription, elongation, and termination. And we'll talk about each of these in turn. So we start again with a double-stranded DNA template shown at the top here. There is a specific sequence where initiation begins. It's called a promoter. There is a specific termination sequence shown in red here. The transcriptional machinery, which we'll talk about in a moment, which includes the enzyme Pol2 as well as many other proteins, binds to the promoter, and that is the initiation step. And we'll talk exactly about what happens during initiation. This is the primary step for regulation of transcription. We'll talk a bit about how that works as well. It's important to note that many initiation events abort. They don't go farther than the production of a short mRNA. Some of those that do not abort, which go on to begin elongation, enter the elongation phase, where the polymerase then moves away from the promoter, moves down the template, and starts to synthesize an mRNA. Elongation can also be terminated before the mRNA is produced. But a certain fraction of the polymerases do reach the termination sequence. The transcript and the transcriptional machinery, the polymerase and the other proteins, are released. The Pol can recycle and go back on another template, and now we have our RNA transcript, which can undergo splicing if needed and export from the nucleus. So those are the generic steps in transcription. We call it pre-mRNA because it has to undergo additional processing. And next, we're going to consider what happens specifically during the initiation step.